And so just a couple of things that I want to say sort of at the outset, because we're so early in the quarter. And the first thing is I just want to remind you of what the expectations are for these Zoom meetings. So there's always a topic for the Zoom meeting, and you can see what it's going to be on the schedule that's posted. And my expectation is that you will prepare for these meetings ahead of time. So you'll look up what the topic is, find the preparation page, and watch the video lectures before coming here. Now, I am aware that today is Tuesday, day 2 of the quarter, and so it may not have been possible for you to do that for this chapter, and that's okay. But you do want to make it a point to do this prep moving forward, and you have plenty of time, right? Because our next Zoom meeting is not till Monday, Tuesday of next week. So there's lots of time for you to go and do that prep for next week. And next week we're covering chapter 3, which is all about microscopy and is super fascinating and fun. Okay. So let's just sort of briefly kind of outline the big picture here. What is the topic we're covering? The introduction to the field of microbiology, covered in chapter 1, is all about understanding what microbes actually are, how we define them, how many types there are, what are their distinctive features, like what makes a microbe a virus and a different microbe as a bacterium and a different one as protozoa. What are the differences between them? We're going to talk about all the many reasons why microbes are important to us. We also want to cover some history. How do we know about them? How long have we known about them? Who helped us figure out like some of the major players in the field in history? And what's on the plate today? You know, it's a big field, so we're just going to touch on a few things. So first, let's just define the term microbe. Microbe is a rather sort of large umbrella term, meaning an organism and or infectious agent that is too small to be seen with the naked eye. So, you know, that includes both things that are cellular, okay, things like bacteria, things like fungus, things like parasites. And it also includes things like viruses, viroids, and prions that we would not consider cellular. They're not really living in the sense of cellular life, but they all have something in common, which is that they can replicate and make more of themselves. So here's a couple of examples. This is an electron micrograph that's taken with an electron microscope, so very high resolution. This is not zoomed in very much. If we could zoom in further and get lots of information from this picture, but it's pseudo-colored. That's why these guys have different colors. And we can see several different species of bacteria on a kitchen dishcloth. Here is the intestine of a mosquito that has been infected with the malaria parasite Plasmodium. This is a protozoan parasite. Here are some fungi, some fungal cells, also an electron micrograph, fungi on a medical implant. And of course, our good friend, coronavirus. So it is really important when you're studying the field of microbiology, and this really applies to cell biology too. It is important to try to, as much as possible, ground yourself in the physicality of them and visual pictures and sort of being able to visualize them in your head. And that's because what's happening is too small for us to see. So it's hard for us to intellectually map it unless we have some exposure and we're thinking about it the right way. So these things are tangible, they're real, but we can't see them and we don't encounter them on a regular basis with our eyes. So it's very useful for many people to use animations, okay? And so I'm not going to show this animation in class just in the interest of time, but I highly recommend that you go to this website and use it to, if ever you're studying something, especially in Unit 2, which can get kind of abstract and heady and you're getting sort of lost in the molecules, go to someplace like this and just zoom into the molecular world and start to feel a little bit like, yes, this stuff is real. I can imagine what it looks like. Now, just to give us a sort of numerical sense of scale, different kinds of microbes and how big are they? Well, the largest microbes are eukaryotic microbes, single-celled organisms like an amoeba, which is an unusually large microbe. Technically big enough to be seen with our eye. It would be seen as a teeny tiny little speck. Okay, so we still consider it a microbe. 400 micrometers, so just about under half a millimeter in size. Much smaller on the other side of the eukaryotic spectrum in size are red blood cells. Human red blood cells are the smallest human cells, and yeast cells. So yeast are a single-celled fungus. So they're in-kingdom fungi, they're eukaryotic, but they are single-celled rather than the multi-celled mushrooms that you see in the forest. And these yeasts are quite small as well. They're on the round 10 micrometers. So 100 times smaller than a millimeter. Now, when we get down to bacteria like E. coli and Staphylococcus aureus and all of our other good friends of bacteria, we're talking about sizes that are around 1 micrometer, so 1,000 times smaller than a millimeter. And for viruses, a common size would be about 1/10 of a micrometer, 100 nanometers, or 10,000 times smaller than a millimeter. Okay, so let's think briefly about how long we've known about them. What would you say? How long have we known about the existence of a microscopic world? Put your answer into the chat. How many decades or centuries or years? Put your answer to the chat. What do you think? How long have we known about microbes? I'm just going to wait for everyone to kind of weigh in here. There's no wrong, well, there is a right answer, but I'm going to tell you the right answer. So there's no wrong guesses. Just guess away. Great. Yeah. So before I say how long we've known about them, let's put this into perspective. How long have they been here? They've been here for a long time, billions of years, way, way before we ever existed. So it's only been about 4 centuries that we have known they exist. And it's only actually been about 200 years of time that we have thought that they mattered to us in any way. So now, what's the difference? Well, we know that they're crucial to many, many things that touch human life. Manufacturing, food production, the ecosystem. They, of course, are responsible for a lot of illness and death around the globe. And they really have had a significant impact on human history and our biosphere. And that was going on long before we knew they existed. The other thing that I think is sort of important to recognize and kind of sobering is that they're sort of at the base of all life. If we killed off all microbes just today, just boom, they're all dead, we would not continue to survive. But if we all died, all the macroorganisms that you think of as being so important, like yourselves and lions and tigers and bears, the microbes would continue on, they'd be fine. So that's an interesting thing to think about. It's also far more likely that if there's life on other planets, it's more likely that life will be microbial in a lot of those instances if it's around. Okay, so if you've done the prep, you're going to know the answers to these questions. If you have not, then feel free to take a guess. Like, don't be shy in this class. Whether I cold call you, whether I ask you to put an answer in the chat, it's fine if your answer is not the one I'm looking for, or if it's not technically correct. I don't care. That's not what today is about. I do want you to engage with these questions, and the reason for that is it's part of learning. It will flag your brain to pay attention and will keep you from falling asleep. So here are some scientists in the field, and I'm going to show you something they did, and you're going to tell me who it was. Who of these people is credited with being the first to name cells or give them that name, dub these things under the microscope as cells? Who was it? Excellent. Yeah. Many of you have the correct answer. Some of you took a guess that was not right, but I appreciate your contribution. No matter whether you had the right answer or whether you're about to learn the right answer, doesn't matter to me. I love that you put something into the chat. You're also welcome anytime you're feeling really stumped to just add someone to the ballot that's not on my list. It's going to be wrong, but that's fun. Okay, Robert Hooke is the first to name cells. Who among these individuals is credited with pioneering the use of antiseptics? Yeah. So those of you who are familiar with the product Listerine may find it easy to remember Joseph Lister. He actually used a compound called phenol. If you've ever worked in a lab, you'll know that phenol causes chemical burns. And so we actually avoid putting it on our skin. But at the time, it was a better option than dying of gangrene on the battlefield. He was a battlefield surgeon. And so nowadays, phenolics actually are still in things like mouthwash. Phenolics have been modified so they don't burn you, in case you're wondering if that mouthwash is about to kill you. Who discovered the first antibiotic according to our history and records? Who discovered the first antibiotic? That's right, Alexander Fleming, 1928. What was that antibiotic? Correct. A good friend made by a very friendly fungus called penicillium. Penicillin, first antibacterial antibiotic. All right, who is credited with pioneering vaccinations? If you know a lot about history, you'll know that there's a complexity to this answer, but among the options presented, who is credited with pioneering vaccinations? That's right, it was Edward Jenner. For no credit at all, just bragging points, what was the vaccine that he developed against? What was the disease he was vaccinating against? That's right, Amy. Yeah, it was in fact smallpox. And he used cowpox as the vaccine in a fascinating and ethically dubious experiment. First to observe living cells, technically credited with this, although again, this would be up for debate, but simplistically, who was the first to observe living cells? That's right. It was Antony van Leeuwenhoek, a fabric merchant who just had a hobby grinding lenses for a microscope that he basically invented and had really good resolution and magnification for his time. Of these individuals, who was the one who has provided very strong evidence to support biogenesis and discredit spontaneous generation? That's right. Our good friend, many of you have guessed him for other things already, Louis Pasteur, and you can be forgiven for that because he did a lot of things in this field and in science in general. He was very prolific. So he certainly did contribute to the field in many ways other than this one. All right. Now I have groups of scientists up here, okay? And these are your options, okay? So these are the options. I'm going to show you a contribution that's sort of a big picture contribution, that one of these groups, every member in this group could be argued to have contributed to that bigger picture, okay? And I'm going to cold call you, and I want to remind you before I do that, that there's no stakes here. I'm not going to remember your answers. I'm much too senile and exhausted for that with the two small children. And I am also aware that people can often like blank on things they know. And I know it's Tuesday, so you might not know these things yet, and that's fine. So you can guess, or you can pass. You can always pass in this class when I ask you a direct question. It is absolutely fine. And just know that when you do that, you're being very brave and supporting other people in the class who have anxiety around being called on so that they can feel comfortable doing that. Okay, so with all of that said, I'm going to show you my first. So a group of scientists that debated the origins of life. So whether biogenesis or spontaneous generation is correct. The group of scientists is a group that all in some way, shape, or form contributed to this debate. And the person that I'm going to tap to try and answer this is Alex. Do you have a guess or do you want to pass?
I'd like to pass, please. I haven't gone through the lecture at all.
Thank you for doing that. And everyone in the class appreciates you for doing that. So that is excellent. The next person I'm going to bother is Angie. Do you have a guess or do you want to pass? I'm not able to hear you, Angie. I don't know if it's a microphone issue. So I'm going to go next to Leslie. Leslie, are you able to unmute? Okay. How about Tiffany? Can you unmute for me?
Sorry, can you repeat the question? I already forgot what it was.
Yes, absolutely. So here is something that one of these groups debated the origins of life. And all you have to do is pick one of these groups as your guess, or you can pass if you don't want to, but feel free to guess.
I would say Pasteur.
It did include Pasteur. Now he's in two of these groups, so you have to pick one of them. Which one do you think debated the origins of life?
Let me go ahead and choose. Hold on, you guys, I'm in class. Sorry.
Or if you want to pass and let someone else pick up the slack, because you've already done the heavy lifting of figuring out which two possibly it is.
I would say the second one.
Okay, that's your guess. This is the second one. And before we move on, I saw someone's hand go up. Was it Sarah? Did you want to say what you thought?
Yeah, that's what I was going to say too, was the one that included Freddie and Needham.
Okay, great. So that's both of you guessed this one. That is actually this guy, this group right here. And it was in the roughly a 200-year span. So this is how long they sat around and talked about it, guys. Just think about that amazing thing. So Pasteur, definitely one of the key players here, but he was sort of at the end. All these other guys did a variety of other experiments in the meantime. Okay, thank you so much for your guesses. Always welcome. I appreciate your bravery. Let's move on to the next prompt and see what we got. Proposed and tested for the presence of endospores. So I'm going to ask Ayumi if you have a guess or if you want to pass. I'm not able to hear you. Ayumi, are you able to unmute?
Yeah, I think like Tyndall and Call.
That is correct. Good job. So yes, in the 1800s, it was actually Pasteur's experiment that was puzzling some people who couldn't replicate his results that caused Tyndall to propose the possibility of a heat-resistant microbe and Kohn to find endospores which were heat-resistant. So together they made that discovery, not really together, but just as a kind of bigger picture idea. All right, now which of these groups could be said to have investigated the germ theory of disease? For this one, I am going to ask Megan if you have a guess or if you want to pass.
I know Pasteur was part of it, so I would say Bazzi, Semmelweis, Pasteur, Lister, and Koch.
Great deductions. Yes, in the 1800s was around the time that people were like, oh, microbes can cause disease, and a lot of people started investigating different aspects of that idea. Okay, now we've already talked about this, so I think this will be pretty easy. Just put into the chat which group you guys think could be said to have been among the early people to observe cells. Put into the chat. You are all correct. Yes, it was Hook and Leeuwenhoek in the mid 1600s. Okay. And last but not least, there's this group, Jenner, Ehrlich, and Fleming. Now, we remember that Jenner pioneered vaccination and Fleming discovered antibiotics. Paul Ehrlich developed one of the earliest synthetic drugs in the lab by taking arsenic and modifying it into solvarsan so that he could use it to treat syphilis. So all of them developed treatments for disease, and this was in a 150-year span. So, you know, really at the beginning of the sort of golden age of microbiology. Okay, so you're next going to think about this. You're repeating Pasteur's swan neck experiment. So some of you may remember this from cell biology. This was his experiment that really put the nail in the coffin to prove that biogenesis, life comes from life, not spontaneous generation. Mice don't come out of laundry, for example. Flies don't come out of meat. But when they did his experiment, some people got a different result. Now, if you have done the prep, you may know the answer to this question. What was Pasteur's result? And what was the reason that some people couldn't replicate his data? So if you know the answer to this or any part of this story, go ahead and raise your hand and let us know what you think happened. Okay. Well, when you go to watch the video lectures, assuming that you have not yet had time to do so, which would be understandable on Tuesday, day 2 of the quarter, remember what happened was that he did this very elegant experiment to disprove spontaneous generation. What had happened is that a number of experiments had sort of said, yes, life comes from life in the macro world, right? Flies are actually laying eggs on rotten meat to cause maggots to generate flies in the second generation. It's not that rotten meat just gives rise spontaneously to flies. So that was well established, but people were still skeptical and thought that microbes did not obey the same laws of biology. They thought microbes did actually generate spontaneously. So Pasteur did this very elegant experiment using a swan-necked flask that you may recall, where he allowed it to be exposed to air, but prevented microbes from getting inside, and so proved that as long as the neck of his flask had a bend in it, then microbes didn't grow in his sterilized soup. But if that was broken and things could just fall in out of the air, then the broth would grow things. However, people were doing experiments all over the world trying to replicate this, and sometimes they couldn't do it. Every time they did it, their experiment would get growth in both conditions, the one that wasn't supposed to grow and the one that was. And so that's why John Tyndall proposed that there was some kind of heat-resistant microbe that was growing that hadn't been properly sterilized. So you want to review this, make sure you understand the logic of this experiment when you look at those video lectures. And that's all I'm going to say about it for now. Okay, let's go on. There are 3 domains of life. You would have learned about them in cell biology. I know it's been a minute, but if you remember even some of them, put those 1 or 2 or 3 of those names into the chat. What are the 3 domains of life? All organisms that are cellular are organized into these 3 domains. That's right, Kelly. Yes, Surmeet, you are correct. Yes, Megan. Yes, Sarah. Yes, Emily. Yes. Okay, so it is bacteria, of course, which is going to be the main focus of this class. Archaea, which are bacteria like, but they have a couple of chemical differences, but they're very similar otherwise. And then eukarya, which includes us and some microbes. So it's a very diverse group. So here are the 3 domains of life: the bacteria, the archaea. They're so similar to bacteria, you cannot distinguish between bacteria and archaea using a light microscope. When you look at them under the light microscope, they're the same size, shape, they have a lot of the same behaviors, they're very simple, they're both prokaryotic, but chemical differences and genetic differences have become apparent, which caused us to group them separately. And then we're part of animals in the eukarya, but there are some single-celled organisms and some microbes in this group. So hopefully the answer to this question is obvious. Write into the chat which of these groups contain microbes. That's right, all of them. There are microbes in every domain of life. Are there any non-living microbes? If I'm asking the question, the answer must be yes. So these are things that can replicate and that may be infectious, but are not cellular. And so we don't consider them living. What would they be if they existed, which they must, because otherwise why am I asking dumb questions? That's right. That's right. Indeed, viruses, viroids, and prions. So this is a nice little graphic to kind of summarize all the things that we're going to talk about. So we've got our bacteria, which are single-celled prokaryotes, our archaea, which are also single-celled prokaryotes. It's very difficult to distinguish them. Can't really tell the difference just looking at them. You have to do more sophisticated chemical and genetic analyses. Eukarya, which includes us and macroorganisms, but also includes things like single-celled animal-like protozoa and algae, right, that are either unicellular or multicellular, but small, microscopic. And additionally includes fungi, most of which are macroscopic, but the yeasts and some of the molds are microscopic. And over here, we've got our non-living viruses, viroids, prions. Okay, so here is a laundry list of diseases, many of which you will have heard of. And some you might know what type of microbe causes them, but maybe you don't. And maybe you think you know, but you're wrong, which is so fun. So below at the bottom, I've listed 4 different options. There's 8 types of microbes, but I'm just going to say bacteria, virus, protozoan, and other. Okay. And what I want you to do is just have a look at this list and think about what you think is the causative agent. Just take a second to kind of reflect on that. Okay, now we're going to go do the lowest stakes, lowest stress cold calling ever, because none of this is on the test. Okay, so you don't have to know this. And also I'm not going to tell you if you're right or wrong straight away. So the only option here though is you do have to make a guess. You do have to guess. You don't get to not guess because, you know, there's no expectation. I don't expect you to know this, and you don't have to know this for the test. It's not part of the curriculum. So you are required to take a guess, and here are your options. All right, so I'm going to cold call Tao first. Tao, which of these 4 options do you think causes typhoid fever?
I would guess bacteria.
Great.
Not
Okay.
sure.
Next person we're going to ask is what causes salmonella? And for that, we're going to ask Surmeet.
I believe it's virus or protozoan. I'm not sure.
Great. Thank you for that guess. Next, we're going to ask Nikki what you think causes mad cow disease.
Protozoan.
Great. Okay, next we're going to ask Kim what causes malaria.
Virus.
Great. So now there's the flu. I'm going to ask Caddy what your guess is for which of these causes the flu. Are you able to unmute, Caddy? Okay, let's ask Janelle. Janelle, what do you think?
I believe it's virus.
Great. Next, we're going to ask Haley, what do you think causes rabies?
A virus.
Great. Gabby, what do you think causes polio?
It's a really good question. I feel like maybe bacteria.
Great. Emily, can you tell us what you think causes the common cold?
I think virus.
Excellent. Now I'm going to go back to Sarah. What about AIDS?
A virus.
All right, good. Back down to, I'm going to cold call a couple people who weren't able to answer earlier just to see if they're there now. Leslie, are you able to unmute? Okay, let's move on to Amy. What about syphilis? Are you able to unmute, Amy? Okay, I'm gonna do a cold call to Alex again. Oh, Amy wrote in the chat and Caddy wrote in the chat. Oh my gosh, I didn't see it there, guys. Thank you. Caddy had said virus and Amy has said virus for hers as well. Thank you. And I'm sorry I missed that earlier. Appreciate your contribution. Okay, herpes, I'm gonna ask Angie, if you're able to answer in the chat. Angie, I know your microphone wasn't working earlier.
I would say bacteria.
Excellent. Okay, gonorrhea. Ayumi, are you able to answer? Either.
I guess virus.
Excellent. Okay, now for smallpox, we're going to have a raise your hand. Raise your hand, especially if you haven't had a chance to answer yet. Raise your hand if you think what smallpox is, or you just want to take a wild guess. Roll those dice. Leslie, go ahead. You are still muted. Oh, there we go.
Virus?
Excellent. Pertussis, raise your hand if you want to answer for pertussis. Anyone? Emily, go ahead. Pertussis, what do you think?
I think it's bacteria.
Excellent. And tetanus, guys. Tetanus. Who wants to answer for tetanus? Tao, did you want to answer for tetanus?
Sure. I think it's the protozoan.
Excellent. All right. Wonderful. So now let's find out what the answers are. And I just imagine that as you were listening to other people's answers, you might have had, oh, yes, I think they're right, or I have a different idea. But at least you had some idea in your mind as you were listening. So here are the answers. Okay. Just have a look. This is, again, does not on the test. Don't have to memorize this, just for fun. I do have an agenda, so I'm going to give you that and the big picture here in a second. If you were wrong about anything, what was the most surprising thing? Write that into the chat. Write into the chat. I didn't, salmonella or common cold or polio. I was surprised by that. So I'll just read out what people say they were most surprised to learn. Okay, so Tiffany and Angie said mad cow was surprising. Kim says that as well. Megan says that. Sarmeet was surprised by malaria. Alex by the flu. Keep those awesome answers coming. Polio and malaria. Gabby, Caddy says malaria. Tao says AIDS. Most surprising. Janelle says malaria. Mad Cow says Nikki. Kelly says pertussis, and she gives an awesome reason. She says pertussis was surprising because we wear masks, and I usually associate masks with a viral infection. Yeah, excellent. Okay, so I wanted to sort of be transparent and break the fourth wall here and tell you why we did that, because it's not part of our curriculum. I want to keep you awake, and this is fun. But I want to sort of remind you that ignorance is a state, a natural state when you're learning. Like, you don't know everything, and scientists who discovered all these things didn't know the thing they were discovering. So I just want to remind you not to be afraid to not know something, to be honest about that in class. Feel free to take guesses also. This was forcing some of you to just take a wild guess. That's fine when I ask you in class. When you're wrong in class, it means that your brain is going to remember it better. So you're going to learn it better than other people who just are cruising along. So that's my little pep talk. Take it as you will. And I just want to address this awesome point that Kelly made. Yeah, people are often do kind of find surprises when it comes to the virus and bacteria thing, because those are the two most common pathogens. They're not the only ones. The other thing that's surprising to people is that fungi can be infectious and or can be a causative agent. It's not generally speaking very infectious, but it can be a causative agent of things like pneumonia. So there's some surprises. Your assumptions might not always be right. Okay, so we have next, really want to tackle this idea of what are the types of microbes and what defines each group, right? What makes a bacterium, an algae, an algae. So here are the 8 groups that we're going to study in this class. I'm going to give you a little bit of time to, I'm going to give you actually like 4 or 5 minutes to write down or look up everything that you can remember or find out about these groups in terms of, try to keep it simple. Don't spend a ton of, don't stress, but like relatively speaking, how big are they? Is there anything unusual or important about their composition? If it is, it's usually going to be about their cell wall. That's the distinctive part. Are there any particular structures, like do they have organelles or not have organelles? Do they have a nucleus or don't have a nucleus? And are they considered living or non-living? So just quickly jot down some notes about the ones on this list that you remember. I'm going to give you a little bit of time to do that. Like I said, about 4 or 5 minutes. And then we will do a brief amount of cold calling, nothing too strenuous, and just kind of review this information, give you a sense of what you need to know. Okay, so see you in 5. All right, welcome back. So I am going to just cold call some people in the room to briefly say whatever they know about each of these groups. Okay, and then we're going to review what all of the information is. And I'll usually, after someone has talked, I'll give the class a chance to add anything that they know that may have been missed. All right, so for bacteria, where we will begin, I'm first going to ask Surmeet what you know about bacteria. Remember, you always are free to pass.
Single-celled in prokaryotes definitely does have a peptoglycan as a cell wall, which also do multiply by binary fission and may or may not be able to be motile because of flagella, may or may not.
Great. Does anyone have anything they want to add? If you do, just raise your hand briefly. Anything that didn't get stated or that you want to say as well? Okay. Next, we're going to go to Nikki for archaea.
I don't know too much. I just know they have ribosomes and a cell wall.
Excellent. Fabulous. And those are true. So that's wonderful. Does anyone have anything they want to add to that? Just raise your hand if there's anything that you want to add to that great answer. Sarah, go ahead.
What I have written down for my notes from earlier is they're prokaryotic also, so they don't have a membrane-bound nucleus or organelles. They're very similar in shape, size, and appearance as bacteria. They also multiply by binary fission. They do have rigid cell walls, but they're not comprised of peptidoglycan. I think that's how you pronounce it. And something I thought was kind of interesting was that it said that many of them are extremophils, that they thrive really well in like extreme environments.
Great. Awesome. Let's next go to fungi. Emily, what do you have about fungi?
I have that they're single-celled or they can be multicellular, and then their cell walls are made of chitin. I don't really know how to say that. And then mainly they live on land, and they get energy from degradation of organic materials.
Excellent. Yeah. Anyone have anything they want to add to that great answer? Okay. Rolling my magic 8-ball here. Let's go to Megan for protozoa. What do you know about protozoa? We're getting down into the lower category, so if you had to pass for any of them, you're always welcome to do that.
I have that they usually have asexual reproduction, but some do reproduce sexually.
Excellent. Does anyone have anything they want to add to that? Looks like, oh, Tiffany, go ahead.
They're single cell organisms and they can come in different shapes.
Yes, that is also true. Excellent. All right, for algae, let's ask Gabby what you have.
I have that they can be single or multicellular. Their cell wall has cellulose. They primarily live in water and are photosynthetic.
Excellent. Anyone have anything they want to add to that great answer? Okay. So now we're in the last three, sort of the non-living guys. For this one, I would like to do it as a raise your hand to take the pressure off of anyone who maybe got through the first few and then didn't quite get to the last three. So if you have answers for viruses, go ahead and raise your hand. I'll pick somebody. Anything that you wrote down about viruses, go ahead and raise your hand and share with us what you wrote. Let's start with Tao.
So virus is acellular and it needs a host to reproduce. It uses the host's resources and it has a protein coat surrounding the core.
Excellent. Did anyone have anything they wanted to add to that? Emily, go ahead.
The core is either DNA or RNA, but it can't be both. It has to be like one or the other.
Yes. Good. So that's where we talked about yesterday how there's genetic material, but it's only either DNA or RNA, whereas cells have both. Good. What about viroids? Does anyone want to comment on viroids? Leslie, go ahead.
Isn't it pyroids are viruses for plants?
Yes, so they are pathogens for plants. They're not viruses, but they only infect plants. That's absolutely right. Yes, and looks like Kim, did you want to add anything?
I was gonna say the same thing, but they also contain only RNA.
That's right. Good. Yeah, they're a little piece of RNA, and as Kim said, and as Leslie mentioned, they only infect plants. And then what about prions, guys? What would you like to say about that? Raise your hand if you've got a prion, some prion info to share. Gabby, go ahead.
They are infectious proteins. and they disrupt cell function. To me, they are the scariest kind because they're like, as you said in the video, like the zombie causers. So they cause neurodegenerative disease, and they're also resistant to sterilization.
Yes, good job. Okay, so here is like a sort of summary. I would not say it's completely comprehensive, but a brief summary of some of the major points about each of these things. So to kind of help you in your studying, to help you remember who's who and what they all have and what you need to remember for the test. Okay, so we are gonna skip the COVID lookup because of where we are in our time. But when you're going to review these slides in this recording, feel free to use this as a way to kind of spice up your review, I guess. And again, not in our curriculum, you don't have to know the answers to these, but it's helpful to apply these categories. So finding out like what category of microbe COVID-19 is, I think everybody knows this. So before we take our break, which is our next item, let me just ask you to put in the chat what category of microbe COVID-19 is. Yeah, or causative agent of the disease. So the virus which causes it is called SARS-CoV-2, and this is the disease it causes. Now, in terms of features that make it part of this group, that would be a fun thing for you guys to look up. Maybe you can take some time on the break if you want to, not necessary, but thinking about what defines a virus, and then how does SARS-CoV-2 fit into that definition? Okay, so what we're going to do after the break is called the What's This game. If you're retaking my class, you've played this before, so you might know the answers, but we're just going to, again, this game is intended to sort of help us to apply our knowledge, like thinking about why it's important to know what these categories are. How is that helpful? And what kind of questions do we need to ask if we're trying to figure out what's causing a disease? Okay, so we're going to take our break right now. I will see you in 10 minutes, which will be 6:05. 6:05, I'll see you at 6:05, and we'll talk about this little application that we're going to do to help us remember and understand the importance of these features of microbes. So see you at 6:05. Enjoy your break. Okay, welcome back, everyone. So I'm going to tell you a little story about a soldier stationed in Africa who got a skin ulcer. And based on what was possible in the area, the two possible diagnoses that the army doctor came up with were either cutaneous leishmaniasis or cutaneous anthrax. Cutaneous, if you didn't know, is a word that means on the skin. So I'm going to tell you some info about these two diseases. Okay, so here's his skin ulcer. Here's some info about cutaneous leishmaniasis. It's caused by a species in the protozoan genus Leishmania, which is caused by sandfly bites. It's carried in the saliva of the sandfly. And cutaneous anthrax, caused by the bacterium Bacillus anthracis, and most common origin would be handling livestock skins, like goat skins. So what I want you to think about is, first off, straight off the bat, which one's your guess and why? Okay, I'm going to give you a second to think about it. And then I'm just going to, I'm going to cold call some people for your guesses and your logic. Okay. All right, so let's start with Lori. What's your guess and what's your logic?
I will pass.
That's absolutely fine, and thank you for doing it. Yeah, no worries. Let's go to Kelly. What is your guess and what's your logic?
I think I'm gonna guess that it was caused by the sandfly bites. And the only logic I have backing this is it seems much more obvious, like much more like realistic that he got bit by a fly than he was handling goat skins.
Great. Okay. Now I'm going to cold call. Let's see. Kim, what is your guess and why?
Oh, I guess I wasn't muted. I actually think it's the handling goat skins. I don't really have a good explanation as to why, but that's just what I'm leaning towards.
Okay, great. And then let me just have anyone who has a guess and a logic they'd like to share. Amy, go ahead. What is your guess and what is your logic?
I would say the sandflies, just because looking at the wounds that the goat skin has red around the border, and the sandflies, those look more similar.
So visual inspection. Okay, Emily, what is your guess and what is your logic?
Mine, I think it's the goat skins, and I think it's that just based off also how it looks, but I think like the actual wound, it's more of a darker color, and the goat skin ones have that darker center. So I just think it looks more similar to that.
Excellent. Okay, so now we've heard, and all y'all are experts, of course. We've heard all these different diagnoses and different logic, all of it based on just very minimal information that you've had so far. And a lot of it is like either what does it look like or what might this guy have been up to. So what I want you to do is I want you to think about a few things. So I'm first going to tell you a little bit of information. Leishmaniasis is caused by a protozoan. So that's a single-celled eukaryote in the genus Leishmania. And anthrax is caused by a bacteria in the genus Bacillus. I want you to think about what questions clinically, diagnostically you would need. If you were running tests, what kind of things would you need to know in order to be able to diagnose this individual? And the test can't be, is it Leishmania or is it Bacillus anthracis? The test has to be something like, what does this causative agent size, or what is it made of chemically, or what kind of internal structures does it have, or this kind of question. So based on what you have recently learned about the types of microbes, and based on what you know about these two types of microbes in particular, what questions could you ask me that would help you figure this out? So if you have a question that you think would be useful to ask, go ahead and raise your hand and ask me. If I'm able to answer it, I will tell you the answer, if it's within the parameters of the game. Tao, go ahead. What's your question?
I would set in a clinical aspect. If Oma was my patient, I would say, have you handling goat skin recently?
Great. So you would ask him if he has handled goat skins, and the answer is he has. But just to cut to the chase, he's also wandered around in the sand. Okay. Any other questions you guys want to ask? And this would be, again, imagine you're able to take a sample of his tissue and you want to ask like diagnostic questions about the causative agent. Like ask me something that would help you narrow down what is going on here. Sarmeet, go ahead.
Maybe it's irrelevant, but I would ask the patient if he's causing any nausea or fever that would help me to match the things accordingly. Like for example, some viruses affect the brain right away.
Right. Yeah, great. So you would ask symptomatic questions. And so now the tricky part is many diseases have very similar symptomology or overlapping symptomology. So what kinds of questions could you ask of the organism rather than of the patient? Kelly asks in the chat, does the sample of the microbe have a rigid cell wall? The answer to Kelly's question is yes. Now, based on that answer, does anyone want to say what they think this is? I'm just going to quickly lower the hands here, just because I know people have been raising them for other reasons, which are awesome. But if you have a guess as to what it is based on what Kelly has just asked and my answer that you can explain, go ahead and raise your hand. Kelly herself could do this if she wants. or ask me another question. So if you think you know what it is, go ahead and raise your hand and tell me why. Sarah, go ahead. What do you think it is and why?
Did you say it does have a rigid cell wall?
I sure did.
Yeah, so then it would be the bacteria because the protozoa don't have cell walls.
Great. Okay. Let me ask you all as a class if you buy this and if you understand why this is in fact the correct answer. So Sarah said, based on the answer to Kelly's question, which was, does the sample have a rigid cell wall? Sarah says she believes now it is Bacillus anthracis. Do you buy this? Yes or no? Write that into the chat. Perfect. Okay. Yeah, it makes sense, right? So everybody who played perfectly. And I just want to say that the reason I do this particular exercise is because, especially based on what most of you are doing, which is like sort of going into kind of a clinical setting where you're going to care about patients, the tendency when trying to figure out what's going on is to ask questions like, oh, how are you feeling and what have you been up to? And those are important. I'm not saying they're not important. But in this class, we're going to learn things and focus mostly on the biology of the organism itself. And that can tell us a lot. It can be very useful, especially when two different diseases are both equally likely for this, because of what the person's been up to and because of how they're feeling. And so asking questions like, well, how big is it? And what's its chemical composition? And does it have a nucleus? Does it, those kinds of things can be helpful. So hopefully that makes sense. Let's talk about one more here. So Maria, a resident at Washington Oaks, which is a local elder care center, develops pneumonia. We need to diagnose her so we know how to properly treat her. So let me begin by asking you what types of microbes can cause pneumonia. This isn't really strictly speaking part of your curriculum, not this question. You don't have to know this per se. I will answer it in a second. But yes, viruses can, bacteria can. That's not all. There's also one other type of organism, fungus. Yes, so we've got fungi, we've got viruses, we've got bacteria. Those are our options. Now, what kind of question can you ask me in the chat? So don't raise your hand for this one, just ask me in the chat. Ask me a question about the causative agent that would help you to narrow down what is causing her pneumonia so we know what to give her as a treatment. So ask and answer in the chat, or ask and answer, ask a question. Ask a question in the chat. You could ask me, how big is it? You could ask me what its cell wall is made of. You can ask me if it has a cell wall. You can ask me what kind of metabolism it does. You can ask me all kinds of questions about the organism. And I will answer them, and then we'll see if you can figure it out. Does it have a cell wall, Kim says. It does have a cell wall. The answer is yes, it has a cell wall. Kelly says, what is the composition of the cell wall of the sample? The composition is chitin. It's made of chitin. And if at any point you think you know the answer based on my answers, you go ahead and raise your hand and tell us why. Gabby, what do you think?
I think it's a fungus, if I'm remembering my note. It's not right in front of me. But I think they're the ones that had the chitin cell wall.
You are correct. Yes, good job. Does everyone understand why she's right? Fabulous. Okay, good job, everyone. Thank you for playing. Okay, so next I'm just going to ask you this question I asked you before. I just want you to put into the chat one reason that microbes are important. I'll read out the answers really quick, and then we'll sort of review some different things. We talked about this yesterday. If you were able to do the prep for this section, you might have a more sophisticated answer now, or a broader question. Okay, so important to the ecosystem. We need yeast to make bread, and that's a microbe. Helps to degrade materials. Yes, fungi do that. to better understand our bodies, because our cells are microscopic, and of course we also are covered in microbes. It's the foundation of life. Some are good for our health, our normal microbiome. Some are necessary for life. We can manipulate them to help us in so many ways. That is all true. Good job. If we understand how they work and what they're composed of, we can understand how diseases work and how to treat them. It does relate to all of life, because all of life is fundamentally cellular and microscopic. It's important for the diagnosis and treatment of disease. It helps us to develop aseptic technique and prevent contamination during surgery. And in a clinical setting, finding out the type of microbe can help us to use the correct targeted therapy to get someone better. Yes, good job, everyone. Okay, so I do want to say a few things about the microbes in our world. Some of them cause disease, and it is important because they cause a lot of human suffering. But the vast majority of microbes are not human pathogens. That is important to recognize. The ones that are have an outsized effect on us, and we really remember them, right? We know about them, but there's so many microbes that don't have that impact. Some of them are helpful, directly living on us and are part of our immune system, really. Some are important to the ecosystem, doing things biochemically that other organisms are not able to do, such as decomposition of organic materials and also creation of organic materials through things like carbon and nitrogen fixation. Some of them have been manipulated to help us make food or make goods. And some of them have even been engineered, genetically changed in order to produce medicines and to help us with research. Some of them are very useful at being an example of a particular kind of process. And what do you think? Do we know everything yet about microbes? Sure don't. That's right. In fact, less than 1% of all microbial species can be grown in a lab. Even the ones we know about existing, we cannot grow all of them. And since the advent of sequencing, genetic DNA sequencing, being able to read the genome of different cellular organisms, it has really sort of blown the lid off of the biodiversity in the microbial world. Just as one example, in the Sargasso Sea, which is also called the Bermuda Triangle, just a little sample taken and analyzed by genetic sequencing revealed almost 2,000 new bacterial species that had not been previously identified. And this is just one example. I mean, so that there is just a ton of microbes out there that have yet to be discovered and have every one of them potentially utility in the things that matter to us. There are lots of challenges that microbiologists remain to deal with, such as the disease and impacts on the environment. So it is an important field. Now, looking back, understanding microbes has allowed us to do some amazing things, like develop a vaccine that in the 70s basically eradicated smallpox from the earth, which had previously been a very serious, often fatal disease that, you know, would kill people or disfigure them, because if you survive, you often have scars. Also the Black Death, which has been a source of suffering in various times in history, most famously in the Middle Ages, where 1/3 of the European population perished in just a few years, which is caused by a bacterium, has now been well controlled with antibiotics, so it is not eradicated. But despite these milestones and these accomplishments, things like respiratory infections and diarrhea are still the largest causative agent of death in children worldwide. And in the US, there's still plenty of people who suffer and die from an infection. And notably, of course, recently we've had this global pandemic, and we've all been probably paying attention to what the outcome of that has been. It certainly has had a big impact on politics and the economy and people's personal lives and, you know, death tolls and so on. And there are new diseases emerging all the time. Some of them are coming because they have developed resistance to treatments we previously had been using to control them, such as tuberculosis, which is particularly worrisome. And some of them are coming across the species barrier. Okay, so we've already done, we've already done our break. So what we're gonna do now is a Kahoot. Let me pull that up. What did I do with it? I think I had it loaded up and then I maybe like possibly could have accidentally unloaded it. So let me load it back up here in a second. It was all set to go and I don't know what I did with it on my very jumbled up screen here. Here is the Kahoot. Let me start it and then I'll share my screen with you. Okay, so on your screen, you're going to see a number. If you are joining in Zoom for the first time, the way to get to the Kahoot is either to open a new browser window and put kahoot.it in and then put this game pin in, or download the Kahoot app onto your phone or mobile device. However, make sure that if you do it this way with the kahoot.it, that you also keep this shared screen window visible on your computer, because you need to be able to see it at the same time as the Kahoot screen. Okay. Now we have 19 people on the call today. We have 10 logged in. Let's see if we can get everybody loaded up here. Getting closer. So just 30 more seconds and then we'll get started. See if I can get everybody in, or most of the people in. Sometimes it can be hard to get to the Kahoot, or you might get kicked out of it, in which case just know that the pen is always displayed on the lower right of my screen. So you could relog back in. And if you absolutely can't get to the Kahoot, you can always put your answers into the chat. It won't record them in the Kahoot, but this isn't for a grade. All right. We can get one more person in and let's roll. Here we go, guys. Getting started on our Kahoot today. Fabulous. Thank you, Alex. What best describes the contribution of Tyndall and Cone? It was hypothesized or discovered endospores. Good job. What best describes the contribution of this group of scientists, Jenner, Ehrlich, and Fleming? It was developing antimicrobial interventions. Good work. Which group investigated germ theory, the theory that specific microbes cause disease? Yes, it was Bassi, Semmelweis, Pasteur, and Koch. Which domain contains microbial members? Indeed, the answer is all of them. A microbe 100 nanometers wide made of RNA and protein is probably a virus. Indeed, a virus. This is smaller than a cell. There's no DNA, but there is more than just RNA, so it's not a viroid and it's not a prion. A microbe with a cell wall made of chitin is probably a fungus. Yep. 0.45 millimeters is equivalent to 450 micrometers. Congratulations. Last one, true or false? Most microbes are pathogens and cause disease. That is false. Most microbes are not pathogens. Okay, let's see who made it to the podium today. We were the runners up. I know it was a close race. You were all neck and neck. So if you didn't end up on the podium, I know that you were just right behind these fast people, and congratulations to everybody. Good work, guys. Okay. Let's go back to our slides for more fun things that I have to tell you today. All right, so moving on to lab, I have a couple things that I want to talk about. Unknown ID project, just briefly introduce you to that, kind of big picture it, give you an opportunity to review the lab 1 calculation if you need it. And I want to preview the resolving power calculation that you need for lab 2, because you're going to submit your pre-lab you guys are going to submit on Tuesday night. So there is time actually asked on Monday, but you're going to be doing a lot of that reading and stuff before we meet again. Okay, so the Unknown ID project is a long project. It takes a couple or 3 months. And the first part is the easiest, and it's due on Sunday. You go to the wiki page and you just put your name next to an unknown ID number. That connects you to a particular species of bacteria, but you don't know who it is. And I don't know who it is. The only person who knows who it is our technician, To Win, and she is the person you're going to communicate with primarily on this project. So once you have this, you're going to start part 1, and that is going to be to request a gram stain from her, and she'll send you a picture, and you're going to use that to determine the shape of your cells and whether they're gram positive or negative. After that, you're going to use a dichotomous key and work down to a group. So you're going to do 2 or 3 or 4 tests to figure out what your group is. And again, you'll be emailing her, hey, can I get this test? And she'll send you a picture, you'll interpret it. And then you're going to tell me what you think your group is. And then at that point, really the meat of the project begins. You're going to have a group that has a dozen or 2 dozen or 200 species in it, and you're going to have to do testing to eliminate all of the species that are not yours until you have just 1 left. When you figure out who that is, you write your lab report and submit it at the end of the quarter. So do this week on Canvas, sign up for your unknown ID number. The sooner the better. Next week, we're going to talk about how to do part 1. This is technically not due until week 3, but your goal should be to finish it next week. So you're going to sign up on Sunday, and by the following Sunday, ideally, you'll be able to submit part 1 and be done with it so you can move on. Now, I just want to remind you that you learned how to do CFU calculations for air and water samples on Monday. I do have some slides that we can go through if necessary to refresh our memories about these. And so I'm actually going to look at our time. Yeah, we have time to do this right now. I want you guys, I'm gonna just kind of exit out of the presentation part. I want to do like a little improv-y thing here. Okay, so first of all, give me a number that is around 100, but not exactly 100. Put it in the chat. I'll pick one of your answers. 85. I love it. 85. Let's put in 85. Okay. 85. Give me a fraction of a square foot that's not 1/8. Give me something else that's not 1/8. Oh, a quarter of a square foot. I love it. Let's put in a quarter of a square foot. Okay, so let's imagine that the size of this plate was a quarter of a square foot. And give me an exposure time. 30 minutes. Okay, so what fraction of an hour is that? Half an hour. Okay. All right. Okay, so if our surface area is a quarter of a square foot, how many of these plates would we need to sample a whole square foot? We would need 4. Yeah. And so what should we do to this number, 85, to account for the fact that we want to know how many were in a whole square foot, but we only sampled a quarter of a square foot? We have to multiply by 4. Okay. So we could either divide by a quarter or multiply by 4, which is the same thing. Okay, because we only exposed for half an hour instead of a full hour, what do we have to do to this number to account for the fact that we want to know what would have fallen in a full hour? Multiply by 2. Okay, so we can multiply by 2, or we can divide by half. That's the same thing. Either way, calculate the answer and tell me what it is. Let me just change this number too, because we don't want to do this. We don't want to have this wrong. So if we take 4 times 2 times 85, we get 680. Does that make sense? Yes, no, or kinda? The other thing that I've just demonstrated is how to use this template to create your own questions so you can test yourself in practice, okay? If you have questions about how to do this, stick around in the Q&A at the end of class. Quickly, let's do a water sample one together. So I said there's about 30. Give me another number that's similar to 30, but not exactly 30. 27, 19. Let's split the difference and call it 20. Just for the interest of making this really easy to do the math. Okay. 20. All right, so if the volume used was, I'm going to keep this volume because usually we do 0.1 mils, but I gave you this 0.25 mils instead. Just out of curiosity, who knows what fraction of a milliliter 0.25 is? Quarter, that's right. So given that what we want to know is how much stuff would be in a milliliter, but we only used a quarter of a millimeter, we're going to end up multiplying by what to account for the fact that we used a smaller number? We're going to multiply by 4. The other way you can do it, if that's confusing, is you can actually just straight up divide by the volume. You can put it in the denominator of your fraction. 20 CFUs divided by 0.25 mils. We have to account for our dilution factor. What do you want me to do? 10,000 would be fine, but that's a lot. Give me a smaller number than 10,000. That's still a multiple of 10. 100. Okay, let's do 100. Okie dokie, smoky pokey. So that is going to be a smaller number than this. 1, 2, So what is 0.25 divided by 100 is going to be 0.025, right? What's the answer? We can either multiply by 100, or we can do this fancy math. These are both the same. If you're not sure, just pick the one that makes sense to you. What is our answer? Yeah. 20 times 4 times 100 is 8,000. Boom, boom, boom. And there's our answer. That's how much in the original water sample, each milliliter had 8,000 colony forming units. Yes, no, or kind of, this makes sense. Sweet. And if it doesn't, please stick around again. Like I said, at the end of class, I'm happy to go over it and do another example for anyone who needs that. All right. So to test ourselves, we're going to do a CFU Kahoot. Do we have time for this? I'm going to save this for the end if we have time, because I have a couple things I want to say first. All right. Next week, we're going to talk about microscopy. And it's really important to understand a couple concepts such as resolution. Resolution is how much detail you can see. And the best definition in this field is going to be the smallest thing that can be seen. How big is that thing that can be seen? What's the smallest thing that will still show up at least as a dot? Now, when we talk about that, what that resolution is and the quantification of it is called resolving power. Then there are some limits to what different items can see. The human eye can see down to about 200 micrometers. That's 0.2 millimeters. Things that are smaller than that, we won't see as anything, and things that are around the size might be just a little speck. The light microscope is 1,000 times better. It sees things that are as small as 200 nanometers, 0.2 micrometers. But that's still limiting us, right? There are definitely structures inside of cells that are smaller than this, such as ribosomes, and viruses are often smaller than this. All of the filaments in the cytoskeleton of a eukaryotic cell, the membrane itself, you know, these kinds of things are smaller than a light microscope could detect. An electron microscope, of course, is the gold standard in the field, or has been for many years, because it can see well below the nanometer range into the picometer range and see even, you know, strands of DNA. So let me ask you this question. When we do this calculation, and there's a way to calculate it, you have to know for the lab 2 next week. When we do this calculation, we do something called resolving power. We get a number, okay? What is better, to have a resolving power that is a small number or a big number? Right. The smaller the resolving power, the better, the more detail we're going to be able to see. That's absolutely correct. Now, we can calculate resolving power. There is an equation. You have to do this for lab 2. In your pre-lab, if you're here for the evening section, your pre-lab is due on Tuesday night and before Wednesday's lab. If you're here from the morning section, because this was a better time for you to come to Zoom, your pre-lab is due on Monday night. And if you're here from the Saturday section, then welcome, thanks for coming. And your pre-lab is due on Friday. So you need to really understand this like right away. Okay, so here's the equation. To calculate resolving power, we take the wavelength of incident light divided by 2 times the numerical aperture. And wavelength is always given to you, and numerical aperture would always be given to you, so you just plug and play. Wavelength would be the color of visible light. You can see it here on a spectrum, or ultraviolet light, which is a smaller wavelength. Or even the electrons in an electron microscope have a wavelength as well. That wavelength is really small. So when we look at this and we, why isn't my thing? Okay, if we want to get the smallest RP value, what kind of wavelength do we want? Do we want short wavelengths of light or do we want long wavelengths of light? Do we want small wavelengths or big ones? Exactly. To calculate resolving power, we have in the numerator of this fraction wavelength. So the smaller this number is, the smaller that number is going to be. So we want small wavelengths of light. The closer to the blue or violet spectrum, the better, and ultraviolet light would be even better. And electrons have a teeny tiny wavelength, so that would be the best. Now, what is Na? The other part of this equation is Na, and that's in the denominator of the fraction. And the Na is a value that's written on the side of the objective on a microscope lens. And so it's just given to you. If we want a small RP, what do we want our Na to be? Do we want our Na to be large or do we want it to be small? Yeah, that's right. Because Na is on the bottom of the fraction, the bigger this number is, the smaller this one's going to be. So we want our Na to be as big as possible. And this is very close to the upper limit of what you'll see for an Na. It's harder to get bigger than that. We want a larger Na. And the Na is actually empirically derived and it's written on here. And it's basically kind of, you could sort of think about it as being like a proxy for how well this lens can resolve things. So the bigger this number is, the better the lens is. Okay. So looking at our time, we are getting pretty close. We're 5 minutes out. The pre-lab instructions and the background information, you're going to learn how to calculate magnification and resolving power. I want you to just do a really quick analysis of this information and tell me a couple of things. So if there was a microscope, okay, that had a couple of objective lenses, an 80x oil that had an Na of 1.2 and a 100x air objective with an Na of 0.5, which of these objectives do you think would be better to use if you want to get really good resolution? Put that into the chat. Which of these objectives would you pick if you were trying to see the most detail? And for this, since you don't have to be right, and you're just taking a guess, you have to put something into the chat. Just take a guess if it's even just a guess on your part. Okay. Fabulous. Now, if you can justify why you made the choice you made, go ahead and raise your hand. Tell us why you picked the one you picked. What was it and why? Let's start with Tao.
So for the better resolution, we want the smaller wavelength, which is the 80, and the larger Na, which is 1.2.
Great. We do want the larger Na. And just to be clear, 80 is the magnification. It's not the wavelength. Okay. Although you are absolutely right, we do want a short wavelength, and we'll get to that in a second. And thank you for saying that. But yes, the Na is so this is the oil objective, the 80x oil, and this is its Na, and we want it because it's a bigger Na than this one. Okay, so that brings me to my next question, which is which light source we want to use, right? As Tao just pointed out, we want the light source that has the smallest wavelength. So what color is that one? Put your answer into the chat. Yes. Of the two options, yellow or green, we're going to choose the green because it has the smaller wavelength. And remembering what the equation is, RP equals wavelength divided by 2 times NA. If you can figure out what the resolving power of the microscope would be if you were using the ADX oil and a green light source, put that into the chat really quick. RP is wavelength divided by 2 times NA. So what does that give us for resolving power if we were to use the ADX oil and the green light? Put that into the chat. And always use units. What are the units? Good. Okay. So Tao has said 208, and the units would be, so RP is the value, but the units are given in the wavelength. The units are a distance unit, and they are nanometers, correct? 208 nanometers. So you would take wavelength, 500 nanometers, divided by 2 times 1.2, which is 2.4. So 500 divided by 2.4 gives us 208. And here, the math has worked for you so that you can see it. This is just to give you a preview of how to do this calculation, which you will need for pre-lab 2. I'm going to end with a reminder of things that are coming up. So Wednesday night, day 3 of the quarter, the orientation module and its associated activities are due. Quick note that there were a couple of errors people found for me, thank you, in my orientation quiz and my lab safety quiz. How embarrassing. I have fixed those, so you might have to retake them if you didn't yet get full credit. Sunday, a bunch of deadlines. Our first review quiz on this material, chapter 1. Our first lab is due. There's a Lab 1 wiki that you have to contribute to. There's a Lab 1 Dropbox that you have to put things into. There's a post Lab 1 quiz, all having to do with that microbes in our world lab, which is online. You have to sign up for your unknown ID number, and you have to, well, you don't actually have to submit pre-lab 2 by Sunday, though it's a good practice. At least read the background information and instructions so that you're ready to submit the pre-lab by its due date in your Canvas course shell. Before Monday night's Zoom, you must view the microscopy video lectures and slides, do that prep, do the microscopy pre-quiz, and read the textbook. Okay, so we will do the CFU Kahoot another time, or I might just assign it separately outside of class, but we are out of time, guys. So I'm going to stop the recording and take any questions. But before I do, I just want to say one thing for anyone who is in the Saturday lab. A reminder, pre-lab 2 is due Friday. Your first lab is this Saturday. Come to that lab, 9 o'clock in the morning, Shuxon Hall, 139. We are 1 building north of the southeast corner of campus. And your lab 1 stuff is still due Sunday. Okay, now we are really ready to be done. So